I think we can start. Before we start with the presentation and the introduction, I would like to highlight a few housekeeping rules for the webinar. First, please make sure that your microphone and your camera is switched off during the presentation to avoid any disruptions. And second, if you have any questions to the speaker and referring to the content of the talk, feel free to put these questions in the chat box, and the moderator will ask them after the talk to the speaker. All right. Welcome all to today's Applying Ethology webinar. The replication crisis in psychology and other fields has highlighted that many key findings cannot be replicated or at least show decreased effect sizes. Given that behavioral studies in animals are in particular quite noisy, this issue is especially relevant for fields that deal with animal behavior, such as applied ethology. And of course, there's a lot of discussion going around about ways how to improve the robustness and validity of research findings. Our Bernhard Voelkl, our speaker today, is a postdoctoral researcher at the University of Bern, and his work is part of this discussion. In his talk, he will highlight how the heterogenization of study populations can improve the validity of research findings. Bernhard, the stage is yours.
Thanks, Chris, for inviting me to this seminar. Yeah, and thanks for the introduction. So my talk today will be about reproducibility of animal research and how it is related with biological variation. I have chosen here a picture of goats that should somehow be a meme for biological variation. So you see many different colors, but of course the animals can not only differ in colors, but many other traits as well. During my talk, I will not talk a lot about goats. So it will basically about biomedical research, where our study species are primarily mice and from time to time rats. So I will focus the whole time on mouse research, but the basic principles that I want to introduce and talk about are basically the same for all kind of animal research. Okay, so the title of this presentation is actually very similar to a title of a paper recently published a couple of months ago. And basically most of what I'm presenting today is based on this publication, a review article which came out earlier this year, Reproducibility of Animal Research in Light of Biological Variation. So if the connection breaks down or so, or you don't like my way of talking, you can also quit and just read it there. This problem with reproducibility is something which is now haunting the scientific community for quite a while. And in biomedical research, there were basically two key studies published about 10 years ago, which really changed the entire field. One was by researchers of Bayer, the other by researchers of another company, Amgen. And in both cases, researchers from these companies took a bunch of published studies, published in high-ranking journals, studies that were considered key contributions in the respective field of, the one was cancer research, the other was neuroscience, and they tried to replicate all these studies. So they went to the original publication, went through the protocols, and then tried to replicate those studies in-house, so in their facility. Researchers of Bayer tried to replicate 67 studies, it's a neat sample size, and Amgen 53 studies. The really alarming finding was that in most of the cases, they were not able to reproduce the original findings. So yeah, out of this 67 study, the majority could not be, so the red part here are the studies that could not be replicated. So the clear majority in case, both in the Bayer study and in the Amgen study. And it was really shocking news. Sometime later, Monia Baker, a journalist from the journal Nature, made a survey asking 1,576 researchers what they think about the reproducibility crisis, whether they think there is a reproducibility crisis, and should ask them several more questions. And what you can see here is just the main summary of, and this was published in 2016. Basically, 52% of those researchers said, yes, we believe there is a significant reproducibility crisis in science. Another 38% said, yes, there is some sort of crisis. And there were just 10% that were not concerned about reproducibility. Only 3% say there is no crisis, and 7% had, well, basically no opinion on the issue. So from this time onwards, it was basically, when this came out, this was a really highly discussed, and from then onwards, it was basically relatively accepted in the scientific community that we do have a reproducibility crisis. Before that, that was the term itself was rather a rather difficult topic, because there were, of course, people who said, well, it's not good to talk about a crisis of science because this might affect funding opportunities, the attitude of the public towards science, and so on. So we shouldn't well speak of a crisis, really. It's we should tone this down or so. But actually, there is concern that there is, and since this piece by Monja Baker, so it's really clear that most researchers are really convinced that there is a big problem, and then we also should call it by this name. Okay, so Monja Baker also asked a lot of other questions. She asked, well, where do you think is this coming from? Which factors contribute to irreproducible research? And also, how do you think could we improve on this? And I will come back to this in a second. If we think of a typical research study in biomedical research, it works like this. So first, you plan your study, you make your study design, then you develop the reagents, you inject them into a model, you take some measurement, you analyze the results, and then you publish this. Well, I mean, I don't know of any study that really goes that smooth, but at least that's the idea how a study should work. In reality, it's not that straightforward and much more messy. The point I want to make here is another one. So that's basically that if you go back to this list of potential variables influencing reproducibility that Monja Baker presented, you will see that these different presumed causes for poor reproducibility affect basically all aspects of this study pipeline. So there are questions like poor experimental design, which is clearly a fault in the study design, or variability of standard reagents, or faulty measurements, biased analysis, and selective reporting. So all these different steps in this scientific pipeline also can be were thought to affect reproducibility. So, but the interesting point is actually what's not here, and this is there's of all these questions asked and all these reasons given for poor reproducibility, none is actually concerning the model itself. And this is surprising because it seems that this that the animal model should be rather central. And I believe there should be questions in there like, do you think that the validity of the animal model of your biomedical research could be an issue for reproducibility? Or do you think that biological variation of the animal models could affect reproducibility? So these were questions that were not asked. And I believe that these questions are actually rather central. And it's not only my belief, but for the rest of the talk, I basically want to present evidence and also some reasoning why this is the case. So I argue that the lab mouse is not equal a lab mouse. We have phenotypic differences. Mice differ. There are genetic differences for sure. So researchers are sometimes working with different strains. We can have sex differences, can have age differences, and we can have, of course, also some sort of individual differences, or also on the genetic levels. Even, I mean, strains are genetically, individuals of a strain are genetically very similar, but also not entirely identical. So even within a strain, we have individual genetic differences. In addition to these individual, or to these genetic differences, we also have environmental differences. Mice are kept under, well, what people believe are standard conditions, but even though cages might look similar or so, conditions always change. So there are no two mice growing up in exactly the same environment. There are always environmental parameters differing between cages, even within a lab, or between cages between different labs. So there are environmental parameters like the ambient temperature, humidity, light regime, noise, odor. If animals are kept in pairs or groups, then of course the social environment. There's the animal caretaker, which might be a different one every time, and the, well, basically experience, and also mood and temperament of this caretaker. These are all environmental variables for the animal that might differ and that might affect the phenotype. So we have also, and of course, how these environmental variables affect the animal might differ depending on the genetic makeup. So that's the so-called gene-by-environment interaction. Some mice of some strains might be much more receptive to environmental stimuli and respond stronger to environmental stimuli than animals from other strains or other species, as it is. So these are all different reasons why phenotypes differ. That means that the animals that we're using as an animal model for some disease differ and vary. 20 years ago, this was already very well described in a landmark study published by Krabby and colleagues, came out in Science, and genetics of mouse behavior, interactions with laboratory environment. So this was a big comparative studies where researchers basically replicated a bunch of behavioral tests in 3 different laboratories. They used both sexes, so males and females. They used a replica, and they used 8 different genotypes, so 8 different strains of mice, and they had a whole test battery of different tests. So some of these were behavioral tests, like the open field test or the elevated plus maze. They had tests for cocaine addiction. They had a water maze. They had a test for alcohol preference and body weight. So a long list of behavioral and physiological measures, repeatedly measured in 8 different strains, both sexes, at 3 different sites. And the question was, how much variation will we actually find? Is it possible that we can run this experiment at these 3 sites and get the same results all the time? Now, what they try to do is to standardize every aspect of the experimental procedure, every aspect of animal husbandry. So they say, a quote from the paper, "We went to extraordinary lengths to equate test apparatus, testing protocols, and all possible features of animal husbandry, including apparatus, testing protocols, age of shipped laboratory reared mice, methods of time of marking before testing, food, bedding, stainless steel cage tops, and etc. etc." So they really tried to have standardized conditions in all 3 laboratories. I will not show all these results because that would be quite a lot. But I will only focus on the outcome of 1 behavioral test, and also only on 2 variables of this test. The test I've chosen is the elevated plus maze. So for those of you who are not familiar with this is a test frequently used in behavioral neuroscience. You have basically a plus-shaped maze and you have with 4 arms. 2 arms are open, and 2 arms are covered by walls. Then you place a mouse here in the middle of this cross, and you're just observing. Usually, you're recording the behavior. You're observing them, what the mouse is doing, and especially you're measuring how much time the mouse is spending in these closed arms and how much time it is spending in these open arms. In general, it is believed that mice that dare to go into the open arms are less anxious than mice that are spending all the time just in these closed arms. And therefore, this test is sometimes used in neuropharmacology to test anxiety-related drugs. But sometimes it's also just used for basically locomotor activity. So that's the test. And the parameters that we can look at here is total arm entries, so how often the mouse is going into one of these arms, and here the second is time in the open arms. So we have these 8 different strains, and we have these 3 different laboratories, and I will zoom in into some of the interesting results. So if you just take the first results here, what you can see if you look at total arm entries is a difference between strains. If you have this HA strain and the black 6 strain, here the C57 black 6, then you see that independent of laboratory where the study was done, in all 3 cases, you see much more total arm entries in the black 6 strain than in the HA strain. So you have strain differences, which means basically genetic differences in the behavioral response of these animals. What you can also see, if you look here, I tried to highlight this by using color. If you compare these 2 labs, Albany and Edmonton, you see that in every, for this variable, total arm entries in the elevated plasmids, for every strain, you see the same pattern, namely that in Edmonton, animals had more arm entries than in Albany. And of course, this is highly significant difference between these labs. So in addition to these genetic differences between strains, you also have differences between the 3 laboratories. Despite that they tried actually to standardize the procedure and do the same thing in all 3 laboratories. But you see sometimes clear differences. And third, in some cases, you also see something which is called, well, basically lab by strain interaction. If you look at, for example, these 2 strains, the 5HT and the B6D2, F2, then you see that if you look at 2 labs, Portland and Albany, in one case you would basically conclude that first strain or so shows less activity than the second strain, that would be in Albany, while in Portland you would come to exactly the opposite conclusion, saying that actually the 5H and 1B is more active than the second strain. So that's a classical case for a lab by strain interaction. So we have all these 3 things. We have genetic differences, but we also have lab-specific differences, and we have lab by strain interaction. And we have them despite, sorry for that, despite the author's aims to equate all aspects of the procedure, despite their standardization attempts. Now, why is this the case? Why do we see all these differences where we hope to find none? And here I will just introduce a concept that is basically was developed in evolutionary ecology, and basically describes this this phenomenon. It's not so well known in biomedical research, so I spent a lot of time over the last 2 years basically just to bring in this well-known and well in behavioral ecology and in evolutionary ecology well-researched phenomenon, which is called the norm of reaction. The norm of reaction basically says that if you have an environmental parameter, it will influence the phenotype of your animal. It can influence the phenotype of your animal. That means depending on in which environment the animal is growing up, it will in the end show a more or less different phenotype. So animal growing up in this environment here, where you have a certain environmental parameter, think for example of ambient temperature, but it can be anything else, can be absence or presence of predators or so, will show such a phenotype. Of course, there's a lot of phenotypic variation which is not explained by these specific environmental parameters, but the others. And an animal growing up in a different environment might turn out differently. So the reaction norm is just a function describing the relationship between an environmental parameter and the expected value for a specific trait for one genotype. And this is also an important point. For a different genotype, so for example, for a different strain, this norm of reaction, this reaction norm might look completely different. So it's really a genotype-specific function. So we argued already some years ago that basically this norm of reaction might explain the low reproducibility of biomedical research findings, because what we usually have is one study where researchers take a small sample of animals in one laboratory, kept under various and tested under very specific conditions, and then they get a result for a specific trait, which can be, for example, a response to a treatment. Then later on, maybe this study is replicated by other researchers in another lab. But because it's other researchers, it's another lab, there will be for sure environmental differences. Even if people try to standardize the environmental, the testing environment, we have seen in the crab study, it's not possible. There are certain parameters you can never standardize, like odor, social environment, the caretakers themselves and so on. So you will always have environmental differences, and it might be the case that these environmental differences actually result in a slightly different phenotype, developing a different trait. And so if you find actually a difference between your response in lab 0 and lab 1, first response might be that you say, okay, that's a replication failure, we do not get the same result, but actually it might both be valid results. It's just the case that you sampled from different regions of the reaction norm, and therefore you get different responses. Now, how can we solve this problem? The solution we were coming up, or we were basically propagating for quite a few years, is the introduction of multi-laboratory studies. If we make a study just in under one specific set of conditions in one lab, it's very hard to predict whether this generalizes to other labs or to a broader environmental context. One way how we can basically work against this is by making multi-laboratory studies. So instead of conducting an experiment only in one lab, 2 or 3 or 4 labs should team up, each lab testing a few mice and making the same experiment. And then you pool basically the data and analyze them jointly. So this should describe what happens here. So in this case, we have even 5 labs collaborating. That would be a wonderful collaboration. Each lab might differ a bit in this environmental parameter. Each lab's contributing data. So what you get is basically again an estimate for your trait value, and you get also a lot of variation. You get more variation than in a single lab study. So at first this might sound like a bad idea, but this variation is just representing the true variation that is out there in the animals if they're kept under different conditions. The advantage of this approach is if you then try to replicate this multi-lab study performing another multi-lab study, what you get might be a curve like this blue one. So again, you get a lot of variation, but it's much more likely, basically the mean estimate, the overall estimate of the study will be much closer to the previous study than it was in the case of single lab studies, where you just have one lab with very idiosyncratic conditions and then another lab. So in this multi-lab setting, you can expect more variation, but you can also expect basically higher chances that you reproduce the findings found in the first study. So that's the idea. We tried to test this by basically making use of already published studies. So we first made a meta-analysis looking for suitable data published in the biomedical research literature. And then, so basically studies where we found a lot of independent studies where people use the same experimental paradigm, use the same drug treatment in mice and reported the results. And what we asked is, well, looking then at these individual studies, we see a lot of variation. What would have happened if all these people wouldn't have made their studies just on their own, but if, for example, 2 or 3 labs would have teamed up and conducted the study together? And this is what we basically simulated by resampling from this original research data and playing through different scenarios. The one is just a single lab study where we have just 24 animals divided into control and treatment group, and we get certain results. And we see a difference between the control and treatment group, and we call this a treatment effect. Then we contrast this with the multi-lab approach, where we take the same overall number of animals, but distribute them over 3 different labs. And these were randomly chosen from the whole set of different labs ever performing this test or so. And then we say, okay, what happens if lab A, B, and C, they all make the same experiment, and then they report their raw values? Then we take these values, pool them, and analyze them together. What we see is more variation in such a multi-lab study, because in addition to individual variation, we also have between lab variation, but we also get a treatment effect. So when we do this, what happens, you can see here in this first line of figures here. So if you have only a single lab, you will have sometimes reasonable variation, sometimes low variation within a study. So that would be an individual study, sometimes very low variation, sometimes high variation. So quite a lot of variability, and also quite a lot of variability in the mean estimates. If you have combine the results of several labs, 2 things happen. The one thing that happens is that the within study variation of this multi-lab study increases. But the second thing that happens, that the between study variation, if you look at several of such multi-lab study, decreases. And with this decrease in the between study variation, we also see something else happening. Namely, it's more likely that that such a study will capture the true meta-analytic mean estimate. So we have this meta-analytic mean estimate, which is basically based off hundreds of different studies, all basically researching the same question, testing the same drug. So we have pretty good information where the average response should be. And we can see that multi-lab studies are much more likely to actually capture this true meta-analytic mean than single lab studies. So this is basically, you can see this here in this graph here below, where you have the coverage probability, which is the likelihood, the probability that a study actually captures the true mean, the true estimate for the effect size. And if you have only a single study or so, depending on sample size, so the blue line would be 12 animals, orange would be 24, and then gray would be 48 animals. So we stick just with the blue line here with 12 animals. The coverage probability in this case for this specific data would be just 0.6 for single lab studies. But if you make multi-lab studies with only 2 labs, it's already above 0.8. And with 3 labs, it's approaching 0.9. So it's much more likely that you get reproducible results. It comes, of course, at a cost, and the cost is that your overall confidence interval is a little bit wider. So that's the trade-off that you have. You are less certain about your estimate. You have a larger confidence, slightly larger confidence interval, but you're gaining a lot in coverage probability. Okay. The reason why we have this phenomenon that single lab studies are, well, so likely that they cannot be reproduced is something which we call basically this standardization fallacy. It's just this belief that if we standardize something in one lab, we can work with a very small sample, and we do find an effect, but we completely ignore the problem that if this is replicated somewhere else, people might have different standards, or even if some of the standards are the same, there are always differences between the labs, and then it becomes very hard that you can reproduce the original finding. So the standardization fallacy, as formulated by Bernhard Voelkl, is the excessive standardization of every aspect of the study subject, and this will lead to idiosyncratic results that cannot be reproduced under even slightly different conditions. To explain this, I will digress a little bit and bring an example from the social sciences. So if in the early days of sociology, if you made a survey and asked people, gave people a questionnaire, what you could do is go to some college in the Midwest in the US and ask, well, this population of participants about their opinion on something. But what you see, or what this image basically should somehow illustrate, is that in your sample you have not a lot of variation. I mean, there are individuals, but somehow you will notice certain similarities between those. What social scientists started very quickly is that they diversified their study samples. They wanted to have a study samples that really represents the entire population. It did not only want to know about the opinion of certain age group of college students in a certain urban area or so, but they wanted to have a representative sample for the entire population, which might be the population of the United States or whatever. So what they did is they diversified, they stratified, they were seeking to include people of different participants of different genders, of different socioeconomic backgrounds, and so on and so on. So this is what they did, as they noticed that, okay, our sample is not that representative. Now, what biomedical researchers were doing in their research strategy was actually exactly the other thing. They would pick out one individual, for example, Bruce, and then say, okay, what do we do? We basically try to get a lot of Bruces that are as similar as possible, and then we give each one a questionnaire. And we get a nice sample, and we get very little variation, so we get very precise results. But as you can imagine, these results are rather useless if you want to predict the consumer behavior or the voting behavior of any other person that is not Bruce. So this is the whole essence of basically, or the question of diversifying versus standardizing. And it wasn't actually Bernhard Voelkl who brought this up first, but Ronald Fisher, who already stated, I'm highlighting the most important sentence, which is a highly standardized experiments supplies direct information only in respect to the narrow range of conditions achieved by standardization. So this is exactly what I was talking about. If you standardize too much, if you have just one specific phenotype, you get a question or you get an answer to your question, but this answer might only be valid for this one specific phenotype. And standardization, therefore, weakens rather strengthens our grounds for inferring a likely result. Because in the end, we do not only want to know what Bruce thinks, but we want to know what the population, what the population in general thinks, or when we are working with animal models, we do not only want the response of this very specific mouse tested under this very specific and kept under these very specific conditions, but we want to have more general results. And in fact, as the factorial arrangement well illustrates, we may, by deliberately varying each case, some of the conditions of the experiment achieve a wider inductive basis for our conclusions without any degree impairing the precision. So yeah, the really fascinating thing is that Fisher not only recognized that standardization is a key problem if you want to generalize real results, but basically he also delivered a solution, because what he did then is he developed statistical techniques specifically for this case, where you introduce more variation in your study, where you try to not to standardize, but to diversify your samples, to stratify your samples. So this is the reason why he invented ANOVA. His motivation for inventing ANOVA was basically to be able to heterogenize study populations instead of standardizing this. Okay, so that was basically the take-home message of this presentation. Going back to Fisher and Voelkl, and of course, all the Bruces involved in these studies. Thank you very much for your attention, and I'm here to answer questions.
Thank you, Bernhard, for this very clear and straightforward presentation. We're happy to get questions in the chat box, and as long as people are writing them, I just have one at the very start. In applied histology, people often collect data on farm. So researchers going on different farms collecting data from different animals. Most often, or often it's the same researcher, but also different groups of researchers. Would you consider this as an per se multi-lab study already? Or if not, what can be done to increase or even more increase the heterogeneity of this approach?
So you're going to different farms. This is definitely a step towards a multi-lab study. And having various coders coding the behavior or so is also something introducing heterogeneity in your study, and is therefore also useful.
Okay, thanks. So the first question is, the first question from the audience is from Sarah Hinze. Hi Bernhard, thank you for your good and important talk. I'm wondering about where we are in terms of the practical implementation of multi-lab studies. These studies, even so highly desirable, need much more organization, time, and so on. So what is needed for implementation is support from journals, funding agencies, etc. Do you experience such a change in biomedical research? If yes, I think it would be great to share your ways of convincing the different players so that we can help make change in animal welfare science as well.
I was first a little bit distracted because the voice wasn't really Sarah Hinze's voice. Okay, the question is, yes, of course, multi-lab studies are much more work. It's tedious you have to coordinate with others. First, you have to convince others to collaborate with you. You have to find the funding. So every partner in such a multi-lab study has to find the resources and the funding to do the study. You have to quarrel with your collaboration partners about who is going to be the first author of this joint publication. It's much more, there are more bureaucratic hurdles. if all these labs are placed at different, in maybe even different countries, and have to independently apply for experimental licenses. So it's really much more work. So do we really gain so much, and in which way do we gain? I mean, the one thing is really that by doing a multi-lab study, we are increasing the variation of our study animals, and that might help finding more robust effects. But maybe it's possible to get this effect in an easier way. Can we mimic a multi-lab study in a single lab? So I just introduce variation in my lab. I still have the advantage of having more heterogeneous study subjects, but I do not have to quarrel around with all the other institute heads and researchers involved in a big multi-lab study. So that would be actually a nice thing if we can really mimic a multi-lab study in a single lab. Unfortunately, we usually do not know really what are the differences in the lab between the labs, or which variables really to play around with in order to increase the variation. And the other, so this speaks a little bit against this idea of having just heterogenization within one lab. Another thing is that there is one additional advantage of making a multi-lab study. Of course, coordinating with your colleagues is very difficult and time-consuming. On the other hand, it means that you have to be much more precise in the formulation of your study questions. You really have to flesh out your study and define everything in very detailed terms, so that the researchers in the other labs can do basically the same experiment. So there's a lot more also work to be done in terms of communication in order to explain to the others what you're doing. And this forces also you to be much more precise in planning your study. So there's definitely a gain in the long term, because multi-lab studies are usually much more thoroughly planned and much better documented than individual studies. And it's also, if there is, I mean, we have many studies that are somehow flawed because researchers didn't think of that. And that's also more likely to happen if you have single lab studies, because in multi-lab studies, you have multiple PIs, everybody checking the experimental protocol, and you have just more, the more people are really checking this, the more likely it is that you will spot any flaws in the experiment early on, which means in the long term, it can save you a lot of trouble. There was one more point I want, one more answers. Do we already see changes? Yes. In biomedical research, there are now the first programs rolling out. So one was, actually there is 3 I'm aware of. The one is the Brazilian consortium, where they're making replication studies of earlier Brazilian research findings. And all these replication studies are multi-lab studies, and they try to make at least 100 of these replication studies. So that's a really big effort. It's just in the making, so I think they have started with the first studies, but results are not out yet. Then there was a second one, which was this cancer project, where they also aimed at reproducing 100 studies in cancer research. In the end, what they achieved was the results were much more modest. So they could only replicate, well, I think it was about not even 30 studies or so. Well, there is still a lot of discussion also what they found in. And the third one is currently ongoing in Germany. It's also in biomedical research, where the German Ministry for Research and Education basically funded and invented a new funding scheme for replication studies. And specifically, they want to have multi-lab biomedical studies. In the first round, I think it was, they said something about some 12 to 14 different research projects have been funded, and this is also ongoing. So I guess in 2 years or 3 years time, we might learn how this went and what the researchers report, whether this was really seen as an advantageous strategy or not. So yeah, there are some initiatives going on right now. How effective they are, we might learn about over the next years.
Okay, great for this. Thanks for this answer. There
That was
are right
basically a
now
talk. Sorry.
a sub talk. We have 3 more questions on this. So Julian Hendricks is saying thank you for your presentation and is asking, how do you think that enrichment in lab animal environments affects reproducibility? From what I understand, enrichment decreases standardization, so would it increase reproducibility?
Yes, it should. And this was also, one of Hanno Wirbel's, basically hypothesis that if you keep animals in enriched cages, you should see more variability and more variation and more heterogeneity in the responses. There have been so far, I think, 3 or 4 studies, some in our lab and some in other labs in Münster and in another place. And the outcome of these studies that specifically looked at the enrichment or are rather mixed. So unfortunately, it's I cannot give a clear answer that I say yes, we see the mice in the enriched cages or so. They are much more variable than the mice in singly housed or so. So mixed results, so we cannot really say. I mean, we should also, for me, it's not so clear what we actually should expect. The one thing you can, of course, say mice kept under enriched conditions, they should be more variable because they have a rich environment and they can, well, that might differentiate their behavior and their interactions with the environment. So you should expect more variation. On the other side, it could also be that a very barren environment might also lead to more variation in the sense that it's basically overtaxing the abilities of some animals. And whenever you come basically, when you're keeping animals in an environment that is basically not really suitable for the environment, where the animals cannot really perform all the behaviors that they should be able to perform. This can lead then to also differentiation and increase in behavioral phenotypes, and what we sometimes describe, for example, as stereotypic behavior is some behavior that animals kept under more natural or enriched conditions are less likely to show. But stereotypic behavior also means that they show behavior, and some animals might develop these stereotypies and some these. So if you then look at the population, and some animals might not develop stereotypies. So what you can see if you look at this population of mice kept under barren conditions is actually behavioral variation. Some stereotypies of this kind, some stereotypy of this kind, and some animals with no stereotypies. So yeah, yes, behavioral enrichment might increase behavioral diversity, but the other extreme, if you go down the other way and just offer them a very suboptimal environment, just barren cages also, this can, for other reasons, also lead to an increase in behavioral phenotypes and maybe also to an increase in phenotypes responding to certain drug treatments or so. And that might be the case why the answers, the empirical studies so far performed do not give a clear picture with regard to does environmental enrichment help or not.
Yeah, thanks for this interesting answer. 2 more to go. Alyssa Manes states that wonderful talk. This is definitely a significant hurdle in animal research. In your experience thus far, do you think it would be realistic to have a few core researchers traveling between labs and either training all research staff involved or running the tasks themselves to help eliminate some of the variability?
It could be the case, but I hope it doesn't happen. Because the idea I want to propagate is that actually introducing this variability is a good thing. Because imagine you just have one researcher who is like Santa, able to basically visit all labs at the same evening and perform the experiment. But this researcher is a rather weird personality, working in a very strange way with the animals. What you would get is then evidence for a certain effect in all different labs, and you say, okay, in this and this and this and this lab, everywhere we find this effect, so it must be rather robust and it must be true, and therefore you believe in this. And then you go on with clinical studies, and then you don't find any effect anymore, and you wonder what went wrong. Now, you basically, what you did is you didn't replicate the study in different labs. What you did was pseudo replication, because I mean, from behavioral ecology, you know, this concept of pseudo replication. If you have just one stimulus and you present the same stimulus to all the animals, I mean, the treatment you want to have the same, but in this case you had a confounding factor, which was the personality of or specific way then this one researcher was interacting with with the animals and maybe affecting the way the animals responded to the treatment. And if you have just one type of this researcher or so, then whatever effect this researcher introduced into the system, you have it in all the labs, and this is pseudo replication basically. So your inference of such a system where one researcher either does all the experiment herself or is training all the other people, and they do it all in the same way, is basically that you don't have that you have some sort of pseudo replication at the lab level. So what I would like to see is variation in researchers, a slight variation in also in testing protocols. So what you should standardize is really what you think matters, and everything else should be allowed to vary. And if you still find the same results, despite that it is being done at different places, with slightly different genotypes, by different people, measured with different apparatuses, and so on, then you can say, okay, I'm rather confident this effect is real. If you always use just the same apparatus and then this apparatus later on turns out to be faulty or whatever, everything which you got was just this idiosyncratic result, which was due to some peculiarities of this one experimental setup.
I've one really brief follow-up on this. You already mentioned that it would already help a lot if we would, for example, increase in-lab variability as well. You mentioned that different coders or experimenters can be running the test. Actually, I can think of different times of day, which might increase the variability in the responses as well. Can you drop some more parameters that just would come to your mind in terms of it would be relatively easy to implement and can be done within one lab just to increase the variability?
The first one is, of course, sex. So in animal research, in biomedical research, it's still very, very common that studies are done just with one sex and predominantly male mice. There are now initiatives specifically from funding agencies that require that you're doing basically doing research with both sexes, with males and females. Not in the very early phases, but in the later phases of biomedical research, at some point you have to include both sexes. But it would be good to do this from the start. So sex is definitely a variable where there is already a discussion going on, and where there right now as the community is switching from the classical approach using just one sex to the approach now using both sexes as standard. Next thing is, of course, strain in mouse research, so that you do not work only with a single strain, but with multiple strains, or related to strains that instead of using these inbred strains, you're using outbred strains or even wild mice or so. These are, also parameters. So it's both about the genotype. Concerning the environment, definitely social housing will increase variation. And yeah, I think, that would be already three very important ones.
Great, thank you. So we have a last question from Birte Nielsen. Very timely talk, Bernhard. Is there also work to be done to convince journals, and especially reviewer number 2, that we need less standardization?
We are working on this, and if you encounter a review, if you get a nasty comment from reviewer 2, you can reply to this and just quote the paper I presented at the start. This recent review paper that a couple of researchers, I was also involved in that, so that we wrote and published earlier this year, came out in Nature Reviews Neuroscience, so rather respected journal, should make some impression. There is another one coming out soon, also on the same topic, that we need more, that we need more variation. The problem is, it is accepted when it comes to sex and genotype. It is not as well accepted when it comes to other factors like environmental or experimental or so. But there is literature out there. I can send you more references if you want to convince reviewer 2. When it comes to journal editors and guidelines, this is a different, this is a slightly different issue. And I mean, yeah, it will take some time until basically this becomes the new standard. There are, specifically with trying to to work on reproducibility, people came up with a lot of ideas, and everybody says, okay, first we have to convince editors and funders that they basically change their mind and accept this as the new standard and enforce this as the new standard. I would be a bit cautious here. There was, I don't know whether I should share the screen. No, I can basically tell this. So in this questionnaire by Monia Baker, there was also the list where she asked, well, what should we do to improve the situation? And there were a long list. There was better statistical training, more time for researchers, less publication pressure, and so on. And there was also the question, whether basically journals should change their policy and basically enforce different standards. And interestingly, this was the question where there was the lowest agreement. So most researchers said, no, we do not think that journals enforcing new standards or so will really solve the reproducibility crisis. They thought better statistical training will resolve the reproducibility crisis, but not new standards enforced by journals. Unfortunately, what we see right now is that most of the discussion about how can we resolve the reproducibility crisis is set, well, funders and researchers should enforce new standards. This is exactly the opposite from what the scientific community, or at least a sample of 1,500 researchers from the scientific community, thought would be a useful measure. So I don't think that you really should lobby within funders and journal editors that they have to enforce these standards. Maybe what would be good is trying to convince them that this is now something which should at least be considered. And what we are actually in this opinion paper, what our main conclusion was, is we don't want to have new guidelines and new enforced standards, but what we like to see is that researchers should at least should somehow consider heterogenization as the default option. And what we would like them to do is at least to discuss why they did, if they do not use heterogenization, if they have the standardized setup, to discuss why and to discuss how this actually impacts their inference space. So the only thing I would add is asking researchers always, well, what's your inference space? About which population do you want to make inference? And if they say we want to know something, how this works in mice, then we say, why did you only test like 6 male mice of the age of so and so? If you want to know what whether it works in mice in general, it's clear that with this restricted sample, you have a much narrower inference space than you think. So I think this would be a way forward. Always basically press researchers who are using standardized designs, what's your inference space you can make inferences about? And then they have to admit, okay, that's rather small. And maybe this will help to change the mindset of those people, but it will take a time.
Thanks, Bernhard. I think this was a great advice for the last minutes of our presentation of the webinar. For everyone who's considering this approach in the research and the test design in the future. We are closing the hour now. I, again, would just like to say thank you for this super informative and clear and straightforward talk, Bernhard.
Thank you for inviting me.
And we will have, in 2 weeks, another webinar with Vitor Ferreira, who will be talking about personality and cognition and chicken. So I'm looking forward to see many of you again there. And again, thank you, Bernhard, for participating and for presenting your research.
Yeah, thank you very much for inviting me. It was a pleasure.